Continuing on our discussion, epigenetics is the bridge between genotype and phenotype. It refers to changes in gene expression that are not caused by changes in DNA sequence. During development, epigenetics is vital for creating and maintaining cell identity. These modifications can be stable and even inherited, which influences how a cell and its descendants behave over time. Here are some review questions that you can reflect on to help understand the information in this next section. What is the relationship between the genome and epigenome? How do epigenetic changes influence development? And what happens when they go awry? These questions are essential in both developmental biology and fields like cancer research and regenerative medicine. During early development, cells undergo a wave of epigenetic changes. These will shape their ultimate fate. The modifications can include DNA methylation and changes to histones. They provide a type of cellular memory, ensuring that a liver cell stays a liver cell throughout countless rounds of division. If these changes occur to germ cells, they could even be passed on to the next generation, which would be called transgenerational epigenetics. The epigenome is the combination of DNA sequence and chemical modifications that regulate its activity. While the genome determines what proteins can be made, the epigenome determines which ones will be made. It is dynamic, responsive to both developmental cues and environmental factors. Things like diet, toxins, and stress can influence. There are three major types of epigenetic regulation: DNA methylation, histone modification, and non-coding RNAs. Each mechanism controls gene accessibility and transcriptional activity. These processes are especially active during early development, but they'll continue to shape gene expression throughout life. Let's first focus on DNA methylation. Methyl groups are added to cytosines, typically at CpG islands in gene promoter regions. This addition, catalyzed by enzymes called DNA methyltransferases, generally will silence gene expression by blocking access to transcription machinery. DNA methylation is critical for X chromosome inactivation, imprinting, and suppression of transposable elements. In contrast, histone modifications will alter how tightly, we can see here, DNA is wrapped around histone proteins. Acetylation will open up chromatin, making genes more accessible for transcription. Methylation of histones can either activate or repress genes, depending on their context. These modifications help cells remember which genes to express and which to keep silent. Histone tags serve as signals to the cell about what to do with a given stretch of DNA. Acetyl groups generally will promote a relaxed, transcriptionally active chromatin state, versus methyl groups will often cause chromatin to condense, reducing the accessibility. This delicate balance determines how each cell will interpret the genome. We can see some of the enzymes here that can control histone methylation and acetylation. Histone acetyltransferases add acetyl groups, versus histone deacetylase removes an acetyl group. And we can see the same for our methyltransferases and demethylases as adding or removing methyl groups, respectively. Consider this discussion question then. How could if histone acetyltransferase is more active, will the chromatin be more open or closed? Let's see. Histone acetyltransferase adds acetyl groups. We learned that acetyl groups generally promote a more relaxed state, transcriptionally active state. So if we have more enzymes adding acetyl groups, the chromatin will be more open or relaxed. Then, therefore, will a gene on this chromatin be more or less likely to be expressed? Will be more likely to be expressed. Some other things you could consider is how could environmental exposures, such as poor nutrition, even toxin exposure, alter the epigenetic landscape during development? What would be the long-term consequences? This is the foundation of developmental origins of health and disease hypothesis, which links early epigenetic shifts to chronic disease risks later on in life. Another mechanism for regulation of gene expression is by non-coding RNAs, or ncRNAs. A newer layer of epigenetic control involves these non-coding RNAs, which are RNA molecules that do not code for proteins, but rather regulate gene expression. For example, microRNA, or miRNA, can bind to messenger RNAs and prevent them from being translated, so therefore reducing translation. This fine-tuning is essential in embryogenesis, and it's implicated in diseases such as cancer. So we return back again to the idea that epigenetic mechanisms are both responsive and dynamic. Unlike DNA mutations, which are permanent, epigenetic changes are reversible, which makes them potential targets for therapies, because imagine being able to switch off cancer-causing genes or reactivate a silenced tumor suppressor gene. So there's environmental factors that we can utilize to alter the epigenetics of a person and affect disease outcome. Specifically, nutrients play a direct role in epigenetic landscapes. Components of the one-carbon metabolism pathway, such as folate and methionine, donate methyl groups used in DNA methylation. This biochemical link explains how maternal diet during pregnancy can influence the health and development of the offspring, even at the epigenetic level. And this is showing kind of the connection here. We have our Krebs cycle, or TCA cycle, with some of these components, succinyl-CoA, alpha-ketoglutarate, acetyl-CoA. We're seeing the relationship between some of these amino acids. We're also seeing this is, over here, our one-carbon metabolism with homocysteine, B6, B12, folate, methionine, feeding into this process of getting us methyl groups that then can be added on, potentially, to our DNA and can play a stabilizing role and also can prevent gene expression. So here's a place to apply what you've learned. Folate is a B vitamin that donates methyl groups. Explain why a lack of folate might cause problems in cell differentiation during embryonic development. Or how might a folate deficiency during pregnancy affect fetal development at the epigenetic level? What could this imply then for a public health strategy? These are the kind of questions that lie at the intersection of developmental biology, nutrition, and preventative medicine. We'll stop here. When we come back, we're going to move into our continuation of development from the zygote into a blastocyst.